Okay, so we're going to start with syllabus point number 60. Syllabus point number 60 is the first syllabus point of evolution, and it ties in very nicely with the first ever syllabus point of the whole syllabus of biology. And the first ever syllabus point of biology is the origin of cells. Where did cells first come from? How did cells start existing on Earth? Now, syllabus point 60 is about animal evolution before vertebrates came into existence, okay? So first of all, can somebody please tell me, what are vertebrates? Okay, so we're starting off a little bit easy. I need some engagement, okay? So can somebody just tell me, what are vertebrates? And don't be scared, don't be shy, because you speaking now, right, in a room of like maybe 14, 15 people, it's going to give you, what's it called? Like an extra confidence boost for later. Okay, it's going to give you that experience to talk to the professors later. So do answer my questions, please. And somebody do let me know, what is a vertebrate, because there's 14 people in here. I don't think that there's absolutely nobody that knows what a vertebrate is. So please tell me, what is a vertebrate? Okay, go on.
Is it animals or a large group distinguished by the possession of a backbone? They have backbone.
Exactly. They have a backbone. And we call that backbone like in anatomical terms, in anatomy, what do we call that backbone?
Spinal column.
Okay, yes, the spinal column, the vertebral column, etc. And in this vertebral column, what do we have? What is the vertebral column protecting? Okay, the vertebral column is protecting the spinal cord, right? It basically is a mass of bones that encases the spinal cord. Okay, cool. So, first of all, for us to understand, the evolution of animals, we have to first look at, what does evolution even mean? Okay, so evolution is defined as the change in the properties of groups or organisms over the course of generations. Okay, and this change is important because it allows them to adapt to their environment. So their environment might constantly be changing, but then the animals are also changing at the same time in order to basically live in these environments continuously. All right. So first of all, the first thing that you need to mention when you have this syllabus point in the exam is, how did the cells first come to be? So this is where you're going to start to talk about syllabus point number 1. So first of all, in the primitive atmosphere, in the original atmosphere, before there were any living beings, there was a lack of oxygen. There was no oxygen in the atmosphere. At the moment, you might know that in the atmosphere at the moment, we have about 21% or something like that of oxygen in the air, and then like 70 or 78% of nitrogen in the air. But back then, there was no oxygen in the air. Okay. Now, oxygen is, when you think about the chemical structure of oxygen, and you think back to, like, the electron shells of oxygen, you know that oxygen has got like a total of, what was it? It's got basically 6 electrons in the outer shell. So in order to have a full shell, it needs to have 8 electrons in its outer shell, right? But it doesn't. It only has 6. So because of that, oxygen can be very reactive. It's looking for anything and everything to react with because it's missing those 2 electrons in its outer shell. So if there was no oxygen back then, this meant that other elements in the atmosphere were able... Oh, sorry. Sorry, guys. Can you still hear me? Yep. Hello?
Yep, we can hear you still.
Yeah, we can still hear you.
You hear me? Okay, cool. Sorry, because my cat literally just jumped on me. Sorry. Okay, so I was saying the primitive atmosphere. So because there was a lack of that oxygen, the other elements, so carbon and hydrogen, were free to react together, okay? But they weren't reacting together willingly, okay? And they needed energy provided to them from lightning. So lightning would often occur in the earth back then, and then that would give the energy to allow carbon and hydrogen to react together. Now, this is very important because when hydrogen and carbon reacted together, they made RNA molecules. And we know what RNA molecules are. RNAs are nucleic acids like DNA, and they're important for storing genetic information. Now, another thing, of course, that they're important for is, that genes code for proteins. Genes are the instructions which are going to basically make a protein. Okay, but what was so special about these RNA molecules was not just that they were able to make proteins, but that they could catalyze their own reactions or their own replication, meaning that the RNA molecules were able to quickly replicate, so they could make more and more and more. And this is how we started to make the first few cells. Okay. So as we mentioned, the Earth's atmosphere had no oxygen by itself, like no elemental oxygen in and of itself. So the carbons and the hydrogens were reacting together. And obviously, as you know now, we have oxygen in the air, and things are different. Okay, so where there is nucleic acids, there is going to be proteins, usually. And you know that proteins are macromolecules. They're the building blocks of life, and they contribute to making the structures within a cell. For example, proteins can they can make up the receptors of the cell within the cell membrane, right? And these receptors control what comes in and comes out of the cell, which is very important. So because they were able to make the proteins then, this meant that they made the first protocells. So protocells just means the first cells to be existing. Now, at first, it was literally just molecules of RNA, right? And then eventually, you know, this RNA, right? It needs to be protected because RNA is very unstable, okay? And it can basically break apart very, very easily. And because of this, it needs something kind of like protecting it, like a protective layer. And what had happened was the cell, right, or the RNA was starting to make the proteins and then lipids. And then essentially, lipids came together to form a bilayer, right? Kind of like the phospholipid bilayer that we have now in our cell membranes. So it made like a membrane to go over the genetic information, to protect the genetic information, kind of. But this cell membrane that was first made, so the first ever cell membrane, was very permeable, meaning that it's not like the cell membrane that we have at the moment, because our cell membrane is very highly selective, very highly selective. So it only allows certain things to come in and it's very controlled, right? And that's because we are very complex people or very complex organisms. But life back then, the life forms that were existing back then were very, very simple, okay? And they didn't really have like big complex transport systems, and for that reason, like, they needed things to come in and out of the cell very easily. Okay, so this is when the first unicellular prokaryotes came into existence, and that's how they came into existence. So what is the order of events? So when there was no oxygen available in the air, this led to the formation of the RNA molecules. This led to forming the proteins. This then led to forming the first unicellular prokaryotes. So very, very basic life forms. They didn't really live for very long. And then eventually, at some point in evolution, there was a sudden influx of oxygen that came into the air. Okay, so we said before there was no oxygen in the air, but now suddenly oxygen came into the air. And how did this happen? Well, there's a specific group of bacteria that are called cyanobacteria, and they basically use something that's kind of similar to photosynthesis in order for them to make energy from nutrients. Okay, so these cyanobacteria, something shifted, something changed in them, and they began to produce oxygen. And we are going to talk about that later. So when they started to produce oxygen and they released a lot of oxygen into the air, right? What this meant was that, first of all, it was toxic to, wait, we will mention that, sorry. Basically, where am I? Yes, so when we had like an increase of oxygen in the air, this increase of oxygen in the air was there to sort of prepare for the future life forms. Because obviously, imagine we now as humans or other animals that are currently existing on Earth with us, they are not able to survive without oxygen, right? So imagine a world without oxygen. So basically, oxygen came into the Earth's atmosphere in order to support us, okay? And the addition of oxygen into the air also made it so that now that there's oxygen in the air, we are able to basically get more energy, right, from our nutrients, and therefore we are able to have more complex structures. So we went from being unicellular cells to multicellular cells. Okay, then eventually we had to transition from using RNA as our main source of genetic information to then using DNA. And the reason for that is RNA is very unstable. It's very likely for it to just, break apart. It's very easy for it to become mutated, right? And DNA is not exactly it's not exactly free from mutations. DNA can also become mutated, but DNA is a little bit better than RNA. So in order to go from very basic life forms to more complex life forms, this was very important for us to go from RNA to DNA. Then when we went from RNA to DNA and we became these multicellular organisms, the first primitive animal to exist, and by the way, this is a question that Stefka asked me last year, Stefka being the head of the biology department. She asked me, what is the most primitive animal in evolution? So if you have this question and if you have her, make sure to tell her porifera. And what is porifera? Porifera are sea sponges. So it's pictured right here. So just think of like SpongeBob SquarePants or something, okay? So porifera were the first primitive multicellular animal to exist. Then from porifera, we got jawless fish, then cartilaginous fish, then bony fish, then amphibians, which were the first animals to live on land, and then amniotes, which include reptiles and birds, and then mammals. Okay, so for this exam, I think it's very important when you're covering evolution for you to understand the order of these events. So this slide is very, very important to you, okay? Very, very important. All right, so now the great oxygenation event. So we mentioned that at some point, right, way back, there was no oxygen in the air. There was no oxygen in the atmosphere. But then suddenly, oxygen came into the atmosphere. And we said that that happened because of the cyanobacteria. But initially, cyanobacteria would use hydrogen sulfide. And by the way, cyanobacteria would live in water, and they used hydrogen sulfide as their electron donor in. Wait, guys, give me 1 second. Sorry. 1 sec. Okay. Sorry. So cyanobacteria would use hydrogen sulfide as their electron donor in cellular respiration. So some of you guys may be second year students or upper year students, and you may have covered biochemistry already. And if you're first year students, this would be good for you to know also. But in cellular respiration, we basically are going to go from breaking down the nutrients, okay, and then getting like electrons from those nutrients, and then having oxygen. Basically, we have oxygen at the end, which is like our electron acceptor, okay, and then we use this oxygen and then we release water, okay. So that's what we do at the moment. But cyanobacteria, what they did was they would use hydrogen sulfide as that electron donor, and then they would release sulfur, okay. But randomly, as we said, a change happened in the cyanobacteria, and instead they started to use the water around them as an electron donor instead. And when they used water, what did they do? They released oxygen as a byproduct. So it's literally the opposite of what we do. We use oxygen and we release water as a byproduct when we are forming energy in the form of ATP, right? But cyanobacteria were using water, right, and then releasing oxygen in order for them to generate energy. Okay, has anybody got any questions at this point? No? Okay, we're going to move on. So, the effects of this new influx of oxygen. So, think of it this way. There was a lot of cyanobacteria that were releasing oxygen at the same time whilst they were in the water. And so this oxygen was going from the water and up into the atmosphere, okay? And what this did was, first of all, if there's a group of animals that are used to living life in a specific way, so they're used to living life in anaerobic conditions, meaning without oxygen, right? Getting this new thing suddenly in their environment is going to throw them off, okay? So what it did was the oxygen ended up being very toxic to a lot of the prokaryotes that were living at the time, okay? So it became very toxic to them because this was not something that they were used to. And because of this, a lot of them died out. A lot of those early prokaryotes died out. But as evolution goes, when some groups are exposed to a new change, they will die. And the other groups that are exposed to the change, they will adapt to it, they'll become resistant to it, and they're going to become better. Okay, so some of them survived. So oxygen is a very important part of the ozone layer. As you know, the ozone layer is part of the atmosphere, and it basically protects us from the UV light radiation that comes from the sun. So UV light is very harmful for us. It's very harmful for our DNA, etc. So the oxygen coming into the air, again, as we said, this was a step to basically prepare life to go from water to living on land, okay? Because when these animals are living in water, the UV light radiation is not really affecting them, right? But when you start living on land, that's when UV light starts to become a problem, okay? And you have to remember the order of events here. We are trying to go from living on water to living on land, and at the same time trying to support ourselves whilst living on land, if that makes sense, okay? So using oxygen in the respiratory chain eventually meant that prokaryotic cells could become multicellular eukaryotes, and they were then able to carry out more energy-dependent functions. So they were able to make new structures, new features, etc. So they were basically able to go from, as we said, unicellular prokaryotes to then like multicellular prokaryotes, and then eventually multicellular eukaryotes, which is our goal. Okay, our goal is to make complex animals here. So how do scientists study evolution? Now, this is also important for you guys to know. You should know what LUCA is. LUCA is the last universal common ancestor, okay? And scientists basically use LUCA, which is like, it's a theory, it's like an animal that they believed lived years and years and years ago, okay? They believe it was a prokaryote that was living in the hydrothermal vents in the ocean floor. So hydro means water, thermal means heat. So this area, this ocean floor, was very, very, what's it called? It would be normally inhabitable. For many animals, that kind of situation would be inhabitable, but LUCA would thrive in that situation because LUCA is a archaea. I'm not sure how to say the word, but LUCA is a archaea, and archaeas, basically, that's how they thrive. They thrive by living in inhabitable conditions, or inhabitable conditions to our standards. So areas that are really hot, areas that are really, really cold, areas that are very acidic. You know, that's where they like to live, and that's where they basically do their best. Now, when we're looking at evolution, animals usually fall into three main super kingdoms. So we have either the animal will either be a bacteria, it will be an archaea, or it will be a eukaryote. Okay. So the differences between bacteria and archaea, even though both of them are prokaryotes, okay, the difference is where they live. So bacteria can be found in like soil, in water, in the human body. You know, you can find bacteria everywhere. But as we said, archaea only lives in very harsh conditions or environments. Now, viruses are not part of the super kingdom. Does anybody know why, or does anybody want to guess why a virus is not considered to be a living organism? If you just think about what viruses do. Why are viruses not a living organism, or considered to be? Any guesses, guys? Last chance.
There's no they can't reproduce, no metabolism. They
Yeah.
lack cellular structure.
Yeah. Okay, so basically, yes. So viruses are not considered to be living beings because they can't live independently, right? Viruses rely very heavily on their hosts. They have to invade the host. They have to replicate themselves inside of the host. They're not really able to replicate themselves outside of the host. So reproduction happens whilst they're in a host, and things like that. So viruses are not able to support themselves. So they're not considered to be living organisms, whereas these other animals are. These other animals can reproduce on their own, can find nutrients on their own. Basically, they're able to survive. Okay, so that was just a fun quick little question for you guys. But moving on. So the evolutionary changes that happen to go from prokaryotes to eukaryotes. Now, I think this is the last part. Yeah, this is the last part of the syllabus that I'm going to mention, because this part is also very, very important. Because the whole syllabus point is basically talking about how did we start life on Earth, right? Before vertebrates. What did we have before the existence of vertebrates? And you might be thinking, oh yeah, okay, we had invertebrates, but that isn't the answer, okay? The answer is everything that we've mentioned up until this point. So we started with prokaryotes, very simple, basic prokaryotes that weren't living for a very long time. And then the aim here is to go to become eukaryotes, which are living for a very long time, okay? Or have a better lifespan and are more complex. Now, in order to go from prokaryotes to eukaryotes, three significant things. Technically we can say four significant things, but I didn't write the fourth one here, but I'll tell you. But four significant things happened. The first would be the loss of the cell wall. So usually most or all prokaryotes will have a cell wall. And usually this is so that they can protect themselves, because they're not living in, some of them are living in very harsh conditions like the archaea, whilst other ones like bacteria might not be living in such harsh conditions, but they need that cell wall to kind of support them and to protect them, okay? So eukaryotes, for example, they don't have a cell wall, obviously, except for the plant cells, right, which need the cell wall for support. But animal cells, they don't have that cell wall. And the reason for why an animal cell doesn't have the cell wall is because we have our cell membrane, right? And our cell membrane is very critical for allowing things to come in and out as part of our transport system. Now, if we had a cell wall, then that would be a structure that would be blocking that transport that we'd need. So for example, one cell that really relies on transport or transport exchange would be the nerve cells. Because as you know, the nerve cells, what do? They're conducting nervous impulses, right? So they send a nervous impulse from one nerve to the next nerve. And the way that they're able to do this is through basically polarization and depolarization, meaning that they are going to allow ions like sodium or potassium to go into the nerve cell and out of the nerve cell. Now, if the nerve cell had a cell wall, this wouldn't be possible. The ions wouldn't be able to come in and out, meaning that the nerve cell would not be able to make an electrical impulse, right? So for that reason, we had to first lose that cell wall in order for us to become eukaryotes, okay? Secondly, we had to form a cytoskeleton. Some of you may have done the cytology exam. Some of you may have passed or failed. I don't know. But the cytoskeleton is a very important structure for the animal cells, or for eukaryotes, I should say, because the purpose of the cytoskeleton is kind of to form like a grid, like basically kind of like a structure which is going to keep all the organelles in place. Because when you think about it, when you think about cells are not like what's it called, they're not very stagnant, just like they don't like really just stay in one place, they move around. Like for example, we can think of red blood cells. Red blood cells don't really have organelles, but red blood cells, for example, they would need to move and things like that, right? Or we could think of like some other cells which may need to move around, for example, white blood cells, right? So if they're moving around, they need their organelles to stay in place. And that is the point of the cytoskeleton, to keep everything in place, okay? So the cytoskeleton is made out of actin filaments, intermediate filaments, microtubules, and all of that is there to make it stay in place, make the organelle stay in place. Then we have the endosymbiotic theory of mitochondria. So mitochondria is a very unique organelle when it comes to eukaryotic cells, okay? And it's unique because it's an organelle that has something very special, okay? Does anybody know what's so special about the mitochondria? When you think about the structure of the mitochondria, what does the mitochondria have? Yeah, go on.
It also has a membrane.
Yes, it has a membrane, yes, but something else, something that mitochondria has that, for example, a lysosome doesn't have, or something mitochondria has that...
DNA?
Yes, exactly, DNA. Mitochondria has got DNA, which is very unique, okay? Because the DNA that's in mitochondria, so first of all, we have two types of DNA, right? We have linear DNA, and then we have circular DNA. And usually prokaryotes, they have circular DNA, right? And usually eukaryotes in our nucleus, where we keep most of our genetic information, where we keep our DNA is linear. And you all know this because I'm sure you have seen the structure of DNA, you know, that double helix. So it's linear, okay? But mitochondria has got circular DNA. Even though mitochondria is part of the eukaryotes, it has circular DNA. So this got the scientists to kind of wonder and think, okay, that's a bit strange. So eukaryotes, all of them, they have, as we said, eukaryotes, all of them have linear DNA. So how come mitochondria's got circular DNA? Well, the scientists came up with this explanation for it, where they basically believe that when the cell was in the stage of trying to become a eukaryote, like that middle transition stage, what it eventually did was it came across a bacteria and it ate that bacteria. And when it ate that bacteria, the structure of that bacteria basically became part of the structure of the eukaryotes. And so they basically believe that mitochondria is like a very old, what's it called? Bacteria, basically, that the eukaryotes ate a very long time ago. Okay, so the endosymbiotic theory of mitochondria is basically what I just explained now, and that is important to the change of going from prokaryotes to eukaryotes. Now, the function of mitochondria, obviously, you guys all know, which is that mitochondria basically gives us our ATP, right? Now, the fourth thing that I didn't write, but I wanted to mention to you guys, is that prokaryotes, they don't have a nucleus, as you know, because pro means before, karyo means nucleus, right? So before nucleus. These structures, or these animal structures, not animal structures, but these species, living organisms, they don't have a nucleus, as we said. They just have the area where they keep the genetic information. And this area is called the nucleoid, okay? Now, the eukaryotes, so eu means true, as in like true story, and then karyotes, of course, karyo means nucleus, so true nucleus. They have a nucleus. Now, going from a prokaryote to a eukaryote, of course, we had to develop a nuclear envelope, okay? And the purpose for that is the prokaryotes, they have a cell wall, and their cell wall is protecting their genetic information. But eukaryotes, they don't generally have a cell wall. Like, think of animal cells, they don't have a cell wall. So how can they protect their genetic information? Well, because of that, they had to form that nuclear envelope that still has pores so that they can contribute into for protein synthesis. Okay. So those are the four important changes that happened from prokaryotes to eukaryotes. Now we're finished with this syllabus point. Before we get to the next syllabus point, does anybody have any questions or any concerns? Anything going on? If you want me to explain something again, you can let me know, by the way.
Yes, do we also have to mention core dates in that syllabus point, or?
In this syllabus point, no. I know that the lecture slide, not the lecture slides, the student notes, there's a lot of student notes out there that are available, from previous students, from students that, you know, were studying here back in 2007 and 2016. And back then, their syllabus was actually different to what we have now, right? So they had like different things that they had to cover. But when you look at the lecture slides for this topic, it doesn't specifically mention the core data. We're going to be talking about the core data in the next syllabus point. So for this one, you don't have to mention the core data. The focus of this syllabus point is, how did life start? Like, how did cells come to existence? And then how did they progress after that? Because the first syllabus point, in the first, like in the whole of the syllabus, the first syllabus point is about the cells, how the cells came to existence. And then this one is basically talking about, okay, what happened after? Now that these cells are existing and now that we have these prokaryotes, what was the next step? So that's what this syllabus point basically focuses on.
Okay, thank you.
No worries. Does anybody have any other questions? Okay, I hope that it was clear. Of course, if you're feeling uncomfortable and you don't want to ask me your question out loud, please do write it down somewhere and then message me after, okay? And then I will try to help you. So, syllabus point 61. This is actually a syllabus point that I had in my exam, the second time that I did my exam. And I wanted to mention something very quickly. So with evolution, you might feel like, oh my God, like it's a lot of information, so much information. How am I supposed to memorize all of this? But honestly, please don't worry. Just highlight what you think might be the most important things. Like in the previous syllabus point, the most important thing that I said, that I mentioned, like everything is important, but the most important thing is the order of the events, okay? So if you want to focus on one thing, please focus on that, okay? Now, for this exam, as I said, this was the syllabus point that I actually had. And I expected that she would ask me a lot of things, because what this syllabus point is basically, you're going to talk about the 5 vertebrate classes. Technically, it's not 5 because one of them is kind of split into 2, and we will talk about that. But you're meant to talk about the vertebrate classes, and you're meant to talk about the new things that they developed in evolution to help them in the environment that they were living in back then. Okay, so literally she could have asked me anything that she wanted, but all she asked me was, what are the innovations that came with fish, basically? So what new things did fish bring? But the thing is that fish is divided into 2, we could say 3 main groups, you know. We have jawless fish, so basically fish that don't have a jaw. Then we have cartilaginous fishes that have a jaw. And then we have the bony fishes that have a skeleton made completely out of bone. And then the cartilaginous fishes, their skeleton was made completely out of cartilage. So she could have been specific and she could have said, okay, you know, tell me about the cartilaginous fishes or whatever. But she asked me like a very kind of technically a broad question. So there was a lot of things that I could have spoken about. So don't worry, the professors are going to ask you questions kind of like that. It might become more specific the more that they see that you know the information, right? But in general, to start off with, they'll start you off with like a broad question, okay? So this syllabus point, what did the 5 vertebrate classes bring to the table? When we're talking about evolution, what was so special about them, okay? So the common theory of progression, as we mentioned before, is we start off with our jawless fish. When it comes to vertebrates, we start with our jawless fish. Then we go to our cartilaginous fish, then the bony fish, then the amphibians, which I said were the first animal group to start living on land, okay? So because they were the first to start living on land, you can imagine that there were many changes that came with their structures, you know, because they had to adapt to life a little bit away from the water. They were still dependent on the water because, of course, when you think of amphibians, you should think frogs, right? So frogs are still dependent on water, but they live their adult life. So after metamorphosis, when they become adults, they live their life on land. Then amniotes, which were the first groups, so like birds and reptiles, to live kind of a bit more independently away from the water. And then mammals, like us, okay? So first and foremost, we already spoke about what are vertebrates. So we said vertebrates are animals which have a vertebral column. Okay. Now, in order for us to even go from what's it called? For us to go from the just normal early multicellular organisms like the periphera. So for us to go from like periphera to more larger and more complex animals, what had to happen was something called the 2R hypothesis. Now, you don't have to know the 2R hypothesis in detail. It's not known exactly when this 2R situation happened, but what you do need to know is that eventually, at some point in evolution, the genomes, so basically genome, to define that, genome means set of genes, okay? So the set of genes were duplicated. There were 2 whole rounds of the duplication of these genes, meaning we had more genes, okay, because of this hypothesis, not because of this hypothesis, but because of this adaptation that happened. So now, if you have more genes, it's only logical to think you have more genes and genes code for proteins, meaning, okay, cool, now that I have more genes, I'm going to have more proteins. Now that I have more proteins, I'm going to have more structures. Okay, it's only logical to be thinking that way. So because we had more genes and we had more structures, we had more genes and then more proteins and then more structures, this meant that we were eventually able to become tetrapods. Tetra in Greek basically means 4, pods means legs. So this basically supported the introduction of 4-legged animals. Okay, so now we're talking bigger animals. Now the Chordata, phylum Chordata. So phylum Chordata is basically the, what's it called? The class or the group that all vertebrates belong in. And it's essentially that they have all the same structures at the embryo stage, okay? So they'll have a mouth, they'll have a anus, they will have a pharyngeal slit, which is going to develop later, okay, and can make the thyroid, all right, in some animals. Then we have the notochord, and the notochord is going to later develop to become the vertebral column. We have the dorsal tubular nerve cord, which is going to become like our spinal cord. Then we have our muscle segments, which of course are going to make our muscles later, and then a tail, okay? So most vertebrates, they have this structure, at least in their embryo stage, okay? Now, I was going to say something, yes, okay. Now, what's important to note about chordata is that chordata are deuterostomes. Now, does anybody know what a deuterostome is? Deuterostome or protostome? Does anybody know? No? Okay. Now, a deuterostome is basically an animal which is going to develop its anus first. So in embryonic development, the first thing that it will make is the anus. And a protostome is an animal which is going to first form a mouth. Okay, so the chordata are usually deuterostomes. They are first forming their anus and then later their mouth. Okay, now the class Agnata, this is the first one that we're going to talk about. Okay, and it's the first animal. Yeah.
I'm sorry, but does deuterostome form the anus first and then the mouth, or the other way around? I didn't get it.
Deuterostomes forms the anus first and then the mouth. Yes.
Okay, and sorry, continue. Sorry.
No, sorry. Did you have a question? Did you have another
Yeah,
question?
well, like the other way around. What is that? What's the word for that?
The word for that is protostome.
Okay, all right.
Protostome.
Okay, thank you.
No worries, you're welcome. Okay, so the first group of animals we're going to talk about when it comes to the vertebrate progression is the jawless fish. And just as the name suggests, they don't really have a defined or really a jaw at all. Now, why is this important? Because you have to remember, what we're going to do is we're going to go from sea sponges that basically they filter water in order to get their nutrients. They live in the bottom of the ocean. They, are very simple animals. So we're going to go from those type of animals to predators, you know, to animals that are going to hunt their own food, right? So essentially we're going from bottom feeders to predators, right? So we're going to first start with our jawless fish. The jawless fish is important to know or basically talk about because the jawless fish are opportunistic predators. And opportunistic predators basically means that when the opportunity presents itself, they will then become predators. Now, how will they be able to do this? Well, they have like a cartilage mouth, like a mouth made out of cartilage, okay? And then they have very sharp teeth, okay? And what they do is they attach themselves onto the prey. Usually if it's like a dead animal, then it's easier for them. So if it's like a dead animal, they will attach themselves to that dead animal. Usually it will be like fish, basically. They'll attach themselves and then they will start to suck out the tissues and the nutrients that animal has, kind of like a parasite, okay? Kind of like a parasite. So there's two of them. There's two types of jawless fish that, you know, we should know. So there's hagfish and lampreys. Those are the two types. There's obviously more examples of jawless fish, but these are the ones that the PowerPoints, that the lecture slides mention. Another thing to note is that jawless fish, of course, are called agnata. Anytime you see Greek words beginning with an A, it means that there's an absence of something. So gnata basically means jaws, okay? It means jaws, like the mouth area, the jaw, okay? So a gnata means that they don't have a jaw, okay? So they can be called agnata, they can be called jawless, or they can also be called cyclostomes. So all of them mean the same thing. Now, the jawless fish, they're made out of cartilage. Their skeleton is made out of cartilage. They have gills, okay, in order for them to respire. And some hagfish, or usually all the hagfishes, they are known as slime fish because you have to remember that these animals, they are only opportunistic predators. They are not actually predators of their own right. So they still have to watch out for other bigger animals or bigger fish. So what they do is they are able to produce like a thick kind of like mucus slime so that they're able to slip away from any predators. Now, what are their contributions to vertebrate evolution? So we said, first of all, they were the first animals or the first vertebrates to go from filter feeding, okay, so being like bottom of the ocean and like basically getting their food from like, you know, water, or like basically getting their food from like algae and plants and things like that, to actually eating other animals. They were the first to do this. They also were the first to develop a complex head, meaning that they had a cartilage and a skull, which would protect their sensory organs, so like their eyes, their ears, their nose, etc., and also a pharynx. Now, they also, because they did develop some level of a backbone. We're going to talk a bit more about their backbone later, but they have some type of a backbone, and that's why they're known as vertebrates. And they were the first to form a heart that had two chambers, so the atrium and the ventricles. Before them, the animals before them didn't have a heart that had two chambers, okay? They had hearts that only had one chamber. Do you guys know why it's important that we have 4 chambers? Or why is it important for an animal to have more than one chamber in their heart? No? Okay. Yeah, go on.
To separate oxygenated and deoxygenated blood.
Exactly. So the more complex the animal is, or the larger the animal is, the more likely it is to be using a double circulatory system, so the pulmonary and the systemic, where we have the deoxygenated and the oxygenated blood. So us having the 4 chambers, for example, as you said, is so that we are able to separate or not have a mix of that deoxygenated and oxygenated blood. But animals that are much smaller than us, right, their blood isn't really traveling very far distances, you know, because they're really small. okay? And for that reason, they don't really need a big separation of their heart. But the jawless fish were the first to develop a heart that had an atrium and a ventricle. They also were the first to develop kidneys to help them with homeostasis. So now that was the jawless fish. Now we're moving on to cartilaginous fishes. So basically, when you think of cartilaginous fishes, you should think of sharks, okay? Now, sharks, of course, are these very scary, big animals that have these very strong jaws, okay? So cartilaginous fishes were the first type of fishes to develop that very strong jaw, okay? And because of that, they were able to become very good predators, you know? They were able to hunt out their prey and very easily, you know, rip apart the prey in order for them to digest the prey, essentially. So because of that, they basically developed really stable teeth, as we said, good jaws, and cartilaginous fishes were the first fishes to be able to develop a bony skeleton later on. At the moment, or usually, they are cartilaginous, meaning that their skeleton is made out of cartilage. But they have the ability, they were the first animal group to have the ability to go from having a cartilaginous skeleton to a bony skeleton. And the significance of this, or why this is important, is because when you're living in water, okay, wait, no, actually, that's not how I want to explain it. I want to explain it this way. Okay, so your nose is made out of cartilage, right? Your nose is not made out of bone. And because your nose is made out of cartilage, it's very easy for you to break your nose, right? So when these animals started to live on land, it was important that their skeleton was not made out of cartilage because cartilage is weaker than bone, right? So before they got to the stage of becoming amphibians, which are the first animal to live on land, they needed to go from that cartilaginous or the cartilage kind of structures in their bones, I mean, not bones, but in their skeleton, to then the bony skeleton, okay? Because as we said, bone is stronger. If they had the cartilaginous skeleton, or if we today, if we as mammals, if we as humans had our entire body made out of cartilage, it would be very counterproductive, okay? We would break our bones, not bones, but we'd break our skeleton very, very easily doing lots of different tasks. So when they're in water, it's not very easy for them to break things, you know, because they're kind of like protected by that water. Another reason why cartilaginous fishes have to specifically have cartilage rather than bone is because bone is very heavy in comparison to cartilage. So we said cartilaginous fishes are usually larger fishes, like sharks. If a shark had a skeleton completely made out of bone, then it would sink, okay? Because water is very dense, right? So it would sink in the water, okay? It wouldn't be able really to float or to swim good. Another thing that developed actually within the cartilaginous fish that I didn't mention before is that they started to develop fins, okay? So they developed paired fins, meaning that they developed a fin on one side and then a fin on the other side to help them basically swim, and then to also help them in their function of predation. So the reason I mentioned that is because right here you can see the agnata, the jawless fish. The jawless fish doesn't have any paired fins. You can see the fish that it's basically trying to get food off of. That fish, it does have paired fins, meaning it has a fin on this side and a fin on the other side. But this agnata, it only has a fin at the end, and we call this fin a caudal fin. Caudal means like the tail, okay? So it has like a tail fin. Okay, and that's it. So because of that, they're not really the best of swimmers, but then sharks are better swimmers because they have that. Another thing that the sharks developed was a solid brain case known as the neurocranium, which helped with the protection of the brain and again, the sensory organs, so the eyes, the ears, etc. Their vertebral column, they developed a better vertebral column than the agnata to help with the mechanical support and the spinal cord protection. And they also developed genital tracts to basically help with their excretion and to also help with their external fertilization or external reproduction. Okay, then we're moving on to bony fishes. From cartilaginous fishes to bony fishes. Now, their skeleton is made out of bone. Okay, there's two main bony fishes that you need to know, and these are the ray-finned fishes over here and the lobe-finned fishes over here. Now, the most important one out of these two is the lobe-finned fishes, because if you look closely, okay, it looks like these are fingers. It looks like these are the bones of the fingers, okay? And this is important to note because after bony fishes, we will have the amphibians, right? And the amphibians, they need to be able to walk on land, jump on land, and do all of these things. And so that progression is very important to note. So the bony fishes, the lobe-finned fishes, I mean, they had this kind of skeleton already in place so that evolution would take place in a basically more efficient way and easier way. So the contribution to the vertebrate evolution. So first of all, they became bony. And the thing is, bony fishes are allowed or not allowed, but they're able to have a skeleton made out of bones. And this is because these fishes are usually smaller, but then also because they have something called a swim bladder. And this swim bladder is full up with oxygen. And because of that, it gives them like kind of, what's it called? It gives them like an extra advantage where they're able to kind of have buoyancy, which basically means that they're able to kind of like float, right? Without them basically sinking, okay? So that is why bony fishes are not allowed, but bony fishes are able to have a bony skeleton, right? Because they have that swim bladder. Okay. So another thing to mention is that lobe-finned fishes were the first fishes or the only fishes to develop lungs, okay? So even though they had gills, they developed lungs. This meant that because they developed lungs, the pulmonary circulation would now start, okay? So that double circulation started for the first time in lobe-finned fishes. They would still use their gills, okay? But they would also be using their lungs. Okay, now the class Amphibia. So some lobe-finned fishes, they then transitioned to land, and then, you know, their lobe fins basically developed into limbs so that they were able to move on land. So terrestrial just basically means land, okay? So they belong to a group, they're called tetrapoda, meaning, of course, four-legged. They, and they're called amphibians. Now, the amphibians, we said, they were still kind of dependent on living somewhere close to water because amphibians are not able to successfully reproduce unless they lay their eggs in water. If they try to lay their eggs on land doesn't have the nutrients or the water that the egg really needs, and then those embryos basically would not survive, okay? So the amphibians were still very much dependent on water. They couldn't go too far away from water, but now they were able to kind of venture and walk around and jump around on land. Okay, so what did they bring to the table here? What changed with them? So because they're the first animal to live on land, many things changed. If you're asked this question in the exam, there's an infinite amount of things that you can basically tell the professor, okay? The first obviously being that the fins of the fish, the lobe-finned fishes, remember how we said that they look like fingers, right? They basically developed and actually became fingers, or something that we know as pentadactyl limbs, meaning 5-digit limbs. Okay, so 5 fingers on both hands and then their legs. And this basically helped them with their mobility, so they were able to move and walk around on land. Now, the next thing is actually very, very interesting also, because the fishes, right? Fishes are not able really to move their head in an up and down motion. They're not able to do that. And the reason fishes are not able to do that is because their vertebral column doesn't really connect to like an occipital area, occipital bone, in a way where that joint would be allowed to be formed. But in the amphibians, they developed the atlanto-occipital joint, which basically means, you know, the first cervical vertebra, which is the atlas, so the C1 basically articulates with the C1 articulates with the occipital bone, okay, and then it forms like a joint. It forms like something known as a condyle, and when it forms this condyle, the amphibians are now able to move their head in an up and down motion. And this is very important because now that they're living on land, they kind of also still need to be careful in case there's anything else there. So they need to be able to look around everywhere, right? They need to be able to move their head. They also need to be able to move their head to catch any flying insects, right? Because frogs, for example, they have this very long tongue where they will go and they will catch their insects or their prey that way, right? So they wouldn't be able to do that if that atlanto-occipital joint hadn't formed, okay, which is that connection between the occipital bone and the first vertebral vertebra, so the atlas, the C1. Okay, so their air also became developed a bit more. So we as mammals, as humans, we have 3 parts of our ears, right? We have our outer ear, which is the part that you can touch, otherwise known as the pinna. Then we have our middle ear, which contains our eardrum, right? Which will transport that, the the vibrating molecules. And then we have our inner ear. So the fishes, before the amphibians then, the animals before the amphibians, the fishes, they only had a inner ear, okay? Because air is a better conductor of sound than water is, right? So fishes, how would they know that something is going on around them? Well, they will feel vibrations on their skin rather than actually really hearing something. But now that these amphibians needed to live on land, they needed to actually hear things, and they were able to hear things. And then that's why they developed the middle ear, specifically with the eardrum structure. Okay, so what else? Their respiratory also changed. Their respiratory organs, not organs, but basically fishes, they normally have anterior nostrils and posterior nostrils, okay? So basically they only have nostrils on top of their mouth, like not in their mouth, but on top of their mouth. But the amphibians, they basically moved their nostrils, the back nostrils, so the posterior nostrils, from on top of the mouth to actually inside of the mouth. And when these posterior nostrils were moved to the inside of the mouth, it basically made it so that they were able to breathe through their nose a bit more. Like it kind of created like a nasal opening, if that makes sense. And the lungs in the amphibians became the primary respiratory organs. When the amphibians, or frogs for example, they go from being tadpoles, so basically the larval stage, and they go through metamorphosis and they become like adult frogs, they then only use their lungs. They don't have gills because they don't really need it. But as larvae, they may still have it, okay? So they also developed their salivary glands and their motile tongue. They got a motile tongue because now their diet was going to be different and they really needed to break down these insects properly. They developed a cornified layer, meaning that they had a bit of a stronger skin. And they also developed multicellular glands in order to release, to keep their skin moist, because they also are able, some of them are able to breathe through their skin. So they need to keep their skin moist in order to actually breathe through their skin. And then they had other multicellular organs, or have other multicellular organs, in order to release poisons, so that they can basically ward off any predators, so that predators don't come near them, essentially. Okay. And then because their diet had now changed, their intestines changed, they developed the large and the small intestine for the first time. The large and the small intestine for the first time were seen in the amphibians, okay? And their heart developed they developed 3 chambers. So they basically developed 2 atria and 1 ventricle, okay? Rather than the 1 atrium and the 1 ventricle that the agnata had, or the jawless fish, I should say. So now we got the next group. So these are the amniotes. So the amniotes, they, of course, as you can tell by now, we're going from better to better to better. Like we keep like improving and improving and improving. Now the amphibians, they were restricted. They had to stay somewhere close to water so that they'd be able to reproduce. Now the amniotes were able to get rid of this problem, okay? Because the special thing about the amniotes is that they were able to lay eggs, which are called amniotic eggs, which basically would thrive and survive for longer and better, and wouldn't need the environment of the water in order for the embryo basically to develop. So the amniotic eggs, they have the extra embryonic membranes within the egg, so the chorion, the amnion, the allantois, and the yolk sac. The yolk sac is particularly important because this yolk sac would provide the nutrients that the embryo would need so that the embryo is able to stay in the egg for a much longer time, okay? And then when it's eventually ready, that's when it will hatch, meaning that its chances of survival will increase, okay? The longer that it's in the egg, the chances of the survival would be increasing, basically. Now, the chorion would be for gas exchange. Now, the amnion, which is what the amniotes are named after, is kind of like the amniotic fluid, okay? So it's basically fluid which would protect the embryo from any kind of mechanical disturbances. So like if you were to shake the egg, for example, the embryo would be protected by that amniotic fluid, okay, or any type of movement. Now, amniotes for the first time were also able to thermoregulate. So this meant that the amniotes were the first to become, or not the first, but eventually the later amniotes, not the early amniotes, but the later amniotes were able to become endothermic. Endothermic means that the temperature outside is not bothering you, in the sense that you're able to regulate your temperature outside of whatever the temperature is at the moment. So for example, we as humans, we have, homeostasis, and we have a lot of other mechanisms which help us stay warm in case it's cold outside, or help us to cool down instead of in case it's hot outside, okay? But there are some animals which are ectothermic, which basically they're a bit more reliant on the temperature. So they need to basically, you can think of it as like a phone. You know, like how a phone, like, when you're using the phone, the battery dies down and then you need to charge it again. Like ectothermic animals are kind of like that, like the more that they use their energy, the more they need to be reliant on going back into the heat and getting energy basically from the heat. So basically think of like crocodiles, you might have seen like videos of crocodiles before where they come up on land and then they keep their mouth open and they're just there like in the sun, and they're there because they're trying to get energy from the heat. It's like they're trying to get heat from the sun so that they're able to use what little energy they have, okay? So we spoke about this, and yes, so the amniotes, they led to forming two main groups. So these are called the Therapsidia and the Synapsidia. And the point of mentioning this is because, I need to go over this also. But the point of mentioning this was because mammals are belonging to the group of Synapsidia. But going back to other things that the amniotes contributed to, so they contributed to internal fertilization. So before them, there was external fertilization happening, meaning that the chances of the offspring surviving back then were lower. But now that there's internal fertilization, the chances of the offspring surviving is higher, okay? Now, the amniotes also were able to eliminate the larval stage, and development would happen within the egg whilst the embryo is protected, as we said. The amniotes would also develop a motile head and elongated neck. Again, this would help them with predation, and yeah, essentially getting food. Their skin evolved to become a bit more dry and less permeable, and this was so that we don't lose water, because these amniotes, as we said, they were trying to make sure that they weren't so dependent on water, okay? And because of that, they may need to stay away from water for long periods of time whilst they're on land, and because of that, they need to really retain the water that they have inside of them. They need to make sure that that water is not going away, since they're going to be away from water for a long time. Now, the pulmonary ventilation, so basically them using their lungs in order to breathe, basically improved because the intercostal muscles then developed in the amniotes, which is not something that was really seen. Sorry, it's not something that was really seen in the previous animal groups. The brain developed better over their spine, and then they basically developed their excretory structures and their kidneys. Basically, And then, as you know, the goal of this is to become better and better and better. So basically, they're just improving to be able to live on land for longer periods of time. Now we have mammals. So mammals are unique because they have the mammary glands. No other species or group of animals have got a mammary gland. Now the mammary gland is the gland which releases milk, okay? Now the point of this mammary gland that the mammals have is so that they're able to raise their offspring for longer, and also they're able to give their offspring the best kind of shot at life initially, because they're able to like train their offspring as to how to be a good predator, or how to avoid prey, etc. And they basically give a lot of nutrients, obviously, through their milk. Now, they descend from the group known as Synapsidia, as we mentioned before, and mammals are usually endothermic. So endothermic animals. So as we said, they basically are able to maintain their body temperature regardless of what's happening outside. Okay. And then their teeth also became better, and they developed lots of different teeth, which is also something that we're going to talk about later. But you know, like the the incisors, the canines, the premolars, and the molars. That's what we mean by heterodonty, lots of different teeth. And this is because some mammals have mixed diets, like we as humans. We are omnivores, of course. We eat plants, we eat animals. And because of that, we needed that mix of different teeth to help us really, chew up the food and help us with our digestion. The integumentary system also developed. So basically, we started to develop hair and fur, and this is to help us with our endothermic abilities. Because, for example, when you are cold, your hair is gonna basically go up, and like the hair on your skin, in order to trap heat in, so that you're basically trying to do your best. Your body's trying to do your best to keep any heat in when you're feeling cold. So we were the first to develop hair and fur. And the reproductive innovations. So, as we said, mammary glands significantly increase offspring survival rates. The nasal conchae basically would enhance the respiration and olfaction, so our sense of smell, and our nasal breathing basically improved. Our brain size also basically improved, and our brain, obviously, it got larger, and this was to help us with social interactions, as most mammals are social. So like humans, for example, we're very social beings. We're able to communicate. We're able to have thoughts. So cognition, we have like a good level of cognition. And other animals are also able to do that. So for example, like monkeys are able to form social groups or little families, and things like that. Now the reproductive system also evolved. So animals that give birth to their live young, so for example humans, developed like a uterus and a placenta, and the placenta, of course, as you know, would then help the offspring in the time of the offspring still basically being in the uterus, would help the offspring so that the survival of the offspring would be better. Now, all in all, as we said, the whole point of evolution is to go from animals which are not really thriving, animals which are not really able to live very well for very long, to animals which are able to live for very long. So whenever you're thinking about all these different changes that happened, you should keep that in mind. So those were the first two syllabus points. How are you guys doing? Is everybody okay?
Yeah, all good.
You're good? I know that it might be a lot of information or very detailed, but please, as I told you, I don't expect you to leave this session feeling like you know everything that we spoke about. I don't expect that of you at all, but I want you guys to have at least understood something. I need you to take something away from these PowerPoints, and then to be able to say to yourself, okay, cool, like I've understood something, or at least I'm not scared of evolution as I was before. Okay? Now we're going to cover the next two syllabus points, and I'm going to try my best to be faster. But the next syllabus point is to do with the phylogeny and the development of the skull. So first of all, in order for us to understand any of these topics to do with phylogeny, you have to be able to define first of all, what is phylogeny? So phylogeny, all it is the evolutionary history. So what happened with this animal, then what happened with this animal, and then what happened with this animal, basically the sequence of events that we had just covered. But now it's going to be specific. This slide is going to be specific to the skull, okay? So when we're talking about the skull first, we have to talk about the three main parts that you guys have to know. So the first part is the neurocranium, also called the chondrocranium. And this area of the skull is the one that basically protects the brain and the major sense organs. It's also known as the brain case because, you know, it's like the case that's carrying the brain. It forms the bottom, the back, and the sides of the skull. And initially, it develops as cartilages. And these cartilage structures are coming from neural crest cells and the mesenchyme of the mesoderm. And this is important for you guys to remember, especially when we're talking about development, because this syllabus point doesn't just want you to talk about the animal groups and what they have in their skull, but it also wants you to talk about how their skull has developed, okay? So the neurocranium is divided into 4 groups. So the regio occipitalis, which is at the back, then we have the regio sphenoidalis, regio otica, and regio ethmoidalis. And then the cranial nerves basically are going to go through these sides. A lot of students usually ask me, like, you know, do we have to draw things like in these exams? And not really. You're not really expected to draw things. I mean, if you're the type of student that remembers things by drawing them, then of course, please go ahead and do that, you know, because usually if you do draw something, then the professors can kind of see, okay, well, this student knows this information well enough so that they're able to kind of recreate it in a picture. Okay, so it will be good for you if you're able to do that, but nobody's going to expect you to draw anything for the exam, okay? So, all right. Now the composition of, so continuing on with the composition of the skull. So we spoke about the neurocranium. Now we have to talk about the next part, which is the viscerocranium. Now the viscerocranium is basically everything else. So it's the facial muscles, okay? So it consists of the branchial arches and their derivatives. So the branchial arches basically come from the pharyngeal arches, which we're going to talk about later. But when we're talking about evolution, we're talking about the fishes first, right? We're talking about the animals that live in water first. So bearing in mind the animals that live in water, they have gills, right? So the viscerocranium for the animals that live in water is going to support their gills, right? Which is the area that is going to kind of like take in the water or basically expel out water. And it provides attachment sites for their respiratory muscles, okay, to basically just support that area. Now, the viscerocranium, where does it come from? It comes from the neural crest cells, which can be found in the ectoderm. So we said the brain case or the neurocranium comes from the neural crest cells and the mesenchyme, okay? Viscerocranium, the cells for this, or not the cells, but the cartilage eventually that's going to make up the viscerocranium, is going to come from the neural crest cells in the ectoderm specifically. Now, the composition, why do I keep doing that? Sorry. Now the dermatocranium. So the dermatocranium is something that is specifically usually found in the fishes, okay? So if we look at this image, which is actually also in the lecture slide, it's a very good image because it shows you where the neurocranium is, okay? Then it shows you where the dermatocranium is, and then the viscerocranium. So when you think of us as humans, we have our neurocranium at the top of our head, right? So we just have it in this area, right? And then we have our viscerocranium for the bottom half of our face. But fishes, they have a dermatocranium right over here, as you can see. So all the blue parts are the dermatocranium. Now, it's important for you guys to know where the dermatocranium comes from, or what kind of cells, or basically the existence. Where is this coming from? Well, it comes from the ossification of the dermal scales of placoderms. Now, placoderms are a specific type of fish. Their other name is armored fish. They had a very heavy kind of scales on the kind of surface on their skin, if you want to say their skin. And basically, their scales basically became ossified, meaning that their scales became like bone, like very, very hard. Okay, the scales became harder, and then when these scales became harder, they formed this dermatocranium structure that we're talking about here. So the dermatocranium, it covers the skull roof. Okay, most of the facial bones, as we can see at the front, but not the teeth, and the opercular bones that protect the gills in fishes. Now the opercular why can I not speak? The opercular bones, there we go. The opercular bones are structures which are found inside of the gills, right? Which are going to help the fish basically take in water or expel out water. But we're going to talk about the opercular bones in the next syllabus points, not this one. So what material makes up the what's it called? The dermal scales, right, that we spoke about, or what material makes up the dermatocranium? Well, it comes from the dermatome, which comes from the mesoderm. So far we have said the neurocranium, basically it's made out of mainly cartilage initially, and then it later ossifies as bone, but it's made out of cartilage first, and then the cells that are making that cartilage were the neurocrest cells, as we said, and then the mesenchyme, the cells in the mesenchyme. Then we said that the viscocranium comes from the neurocrest cells, which are in the ectoderm. Then we said that the dermatocranium comes from the dermatome, which is in the mesoderm. Okay, so those are the differences. We are going to skip that, and now we're going to talk about this. So the two big steps in the evolution of the head. So when we're talking about the head, what changed? Two things. The first thing is that the jaws developed, because remember we spoke before about the jawless fish, and we said how the jawless fish developed jaws, and this was to help them with predation, because again, what is the point of the game here? The point of the game here is for us to go from animals which are weak, not able to fend for themselves, can't really do much for themselves, to animals which are top dogs, predators, they can fight, basically, So the invention of jaws is very important for the evolution of the head, as the jaws basically carry, the teeth, and are also important for articulating. Like, for example, right now when I'm speaking, I'm using my jaws a lot, right? Now, the brain case also, the development of the brain case. So as we know, the brain case is the neurocranium, and it's the top part of the head, not just the top part of the head, but the top parts, as I showed you guys before, and it protects the brain, okay? And this is again, the whole point of this is to protect the organism, to make sure that the organism is going to live for longer. If we didn't have a brain case, okay, if we didn't have a brain case, we didn't have like a thick skull kind of protecting our brain, we would be in danger, and we would be in danger very quickly. Just think about the amount of time somebody falls over, okay? Or even think about like pathogens, some pathogens can go through like our skin or something, right? So what would happen in a case like that? Well, basically we would die, right? So the development of the brain case is very, very important to protect not just our brain, but then of course our eyes also, because we have bones around the eyes, and then around the otic vesicles, so around the ears, and then our nose also. So these are our sensory organs, okay? We also developed sensory organs in order for us to essentially, figure out what it is that's going on around us, and then how to deal with that appropriately. Okay, now the requirements for cartilage. Oh, guys, sorry, is my voice cutting? Can you guys hear me?
We can hear you fine.
You hear me fine? Okay. Anybody else? Can everybody hear me?
Yeah.
Yeah? Okay, cool. All right then. If you can't hear me for some reason, please let me know so I can repeat what it is that I've been saying or whatever. Okay, so the requirements for cartilage formation in the skull. So we said the skull, when it's first made, it is made out of cartilage, okay? So and we mentioned that the first vertebrate animal to have or to develop a cartilaginous skull was the jawless fish. So the jawless fish, even though it didn't have jaws, it didn't have specific jaw. No, it didn't have jaws. Yes, even though the jawless fish didn't have jaws, it still had some type of brain case made out of cartilage. And then eventually this cartilage, as we go up, as we basically go up in this little evolution progression thing that we're doing, you're going to see that cartilage is going to ossify, it's going to become harder, and it's going to become bone. Okay, so where does this cartilage come from? So the mesoderm and neural crest are both very important areas for things that are then going to make our cartilage basically in our skull. So the neural crest cells, they're going to migrate when it comes to the embryonic development, okay? And they are going to induce different parts. So they're going to induce different cells. When we say that the neural crest cells are going to induce other cells, what we mean is that the neural crest cells are basically the boss, and the other cells are basically the workers. So they're like kind of telling these workers, okay, hey, you need to go over there and you're going to become this. So you're going to become the cranial skeleton. Then you're going to go over there and you're going to become soft tissues. And you're going to go over there and you're going to start to form parts of the teeth. Okay, so that's what the neural crest cells were doing. And because of this, they were then able to form the anterior part of the neurocranium, so basically the front parts, right? So the otic vesicles, which contain the ears, and then the jaws in gnathostomos. So what are gnathostomos? We mentioned before, agnatha is an animal which doesn't have jaws, and then gnathostomos are animals which have jaws. Now the mesoderm contribution. So the mesoderm has got different sections. So it's got the paraxal and the Semitic section, and both these sections play a role in the development of the skull. So specifically in the development of the chondrocranium. So what is the chondrocranium? The chondrocranium is the neurocranium. It's just another word for it. In the exam, you can either tell them chondrocranium, which is the Greek term, or you can tell them neurocranium, which is the Latin term. I think it was that order. It should be. Okay, so the mesoderm then gave rise to the posterior chondrocranium. So this is the latter part, the back part of the head. So the large part of the otic vesicles, so the back parts of where the ears are going to form, and also other elements in the skull. We're going over that. Okay, now the transition stages from agnatha to fishes. Now we're talking about the actual evolution. So I mentioned this already. I said that we are going to go from a cartilage skull, okay, to a ossified skull, meaning a bony skull, a skull made out of bone. And again, this is to help with that progression, to make sure our brain is fully protected and we are living for longer with less issues. When these animals are living in water, they don't have many problems to anticipate, really. Because of that, they can afford to have a skull made out of cartilage. But once they start living on land, they need to have a stronger structure protecting their brain and their sensory organs. And then the jaws, as we said, they became better, they developed, they became strong, they allowed for efficient feeding and then predation. In the early amniotes, there was sheet-like cartilages within the skull structure. So even though the skull structure would be ossified, parts of it would be ossified and parts of it basically would be made out of bone, there would still be a little part that was found that had a little bit of cartilage, but not the whole thing. Like it had some parts that were cartilages, but it's going to start like after it stops being, no, not after it stops, but after the early amniotes, you have the later amniotes. So the later amniotes are then going to be the animals to have more of a skull, more of a bone skull, or a skull made out of bone. The mammals, they developed a new type of connections between the skull and the muscles to help them have a stronger bite. And basically, this was so that the different feeding habits would be supported. Okay, because as you know, as we said, mammals can have lots of different types of food, lots of different diets. So not just plant-based and not just animal-based, but a mix of both. Now hominids, which are basically humans, we basically had started to develop increased plasticity of the face. So basically we're able to move a lot of our muscles in the face, and we're able to do this for communication purposes. You know, when we're feeling a certain type of way, you know, our emotions can be reflected on our face and things like that. And that is something that is more unique to hominids rather than any other mammal group. For example, cats don't have that what's it called, that plasticity, so they're not really able to move their muscles like that. And yeah, so we mentioned that. Now the pharyngeal arches. So we said that the, what's it called? Before, when we were talking about phylum Chordata, we said that phylum Chordata, they have, what's it called? They have the pharyngeal slits, right? So now we're going to be talking about these pharyngeal arches. So in ancestral aquatic vertebrates, so these are very, very old vertebrates that were living in water, what they would do is they would take in water through their mouth, and then they would basically move this water out through their gills. And when they were doing this, when they were putting in water through their mouth and out through their gills, they would basically take oxygen out of that water. And that is basically how they would respire and basically form, not form oxygen, but form energy for themselves. Now, the gills over there, they needed to be supported, right? They needed to be protected. So they were protected by the branchial arch skeleton and branchial muscles, which would move them, okay? And these were controlled by the branchial nerves. So here is a picture of what this would look like. So this is basically around where their gills are, okay? So in land vertebrates, so some land vertebrates, they still have these arches. Even though we have lungs, or even though those animals have lungs and they use those lungs in order to take in oxygen, they still have these pharyngeal arches. And in some animals, they develop like a closing membrane, kind of go over it. So for example, the amphibians, because as we said, amphibians as tadpoles, they would need those gills, but then when they become, why did I say as amphibians? Sorry, as frogs, which are amphibians, they would have, as the tadpoles, they would have the gills. Then when they become adults in metamorphosis, they're then going to develop something like to go over that neck area so that they don't have those gills anymore because they don't need them. Now, the arches have different names. The mandibular arch specifically would go through the least change, and this arch is the arch which is associated with eating. And yeah. Now the hyoid and the branchial arches. So the hyoid and the branchial arches, they are going to form new structures. They're going to lose their other function, which was to basically support the gills, okay? Because in evolution, as we said, we're going to lose the gills, right? And we're going to use the lungs instead. So the hyoid and branchial arches are going to form new structures, and they're going to form the middle ear, parts of the middle ear, the hyoid apparatus, which is the hyoid bone in humans, which we need, and then the larynx. So it's going to form these structures, and these structures we are going to discuss more in detail when we get to things to do with hearing and digestion and so on. Okay, now the development. Now we're at the last part of this syllabus point. So now what is the development? How did these things develop? So we've already mentioned where the cells came from. So we said that we have the neural crest cells, some which came from the ectoderm, some which came from the mesoderm. So the embryonic development, when it comes to the neurocranium, so this is the brain case, we said that the cartilage is first going to form. The two types of cartilage that are going to form are known as the paracaudal and the precaudal cartilages. And these cartilages are basically going to come together, okay? And they're going to come together and they're going to go together. They're going to come together and they're going to basically go around the notochord. So when they come together, they're going to form the floor of the brain case, so the bottom of the brain case, the walls of the brain case, and then the roof of the brain case. Now, when it comes to the development aspect of evolution, you don't have to know all of this in detail. Obviously, the more information that you give, the higher the grade that you will get, okay? But they don't expect people to study evolution because a lot of students are scared of evolution. So because of that, whatever you do say about evolution will surprise them, okay? And will make it more likely for them to want to pass you, okay? So don't worry about the information. If you feel like it's a bit much, it will be fine because you're not expected to know it all. Okay. Now, the formation. So the neurocranium initially develops as cartilage, as we said, and then it will surround the brain and the major sense organs. It includes structures like the olfactory capsule that we need for the sense of smell, the otic capsule, which we need for the inner ear. And then the cartilaginous elements, as we said, the paracaudal and the precaudal are going to come together and create the neurocranium's walls. Now, the visceracranium, how is it formed? Well, first of all, we said that it's part of the jaws and the pharyngeal arches. Pharyngeal arches, you could say pharyngeal arches, or you can say branchial arches. It's fine. But the branchial arches, they contribute to, when we say branchial arches, actually, we are more so referring to the fishes. So the branchial arches, they contribute to the formation of the visceracranium, and they support the gills. And as we said, they are attachment sites for respiratory muscles, so that their gills are moving kind of like their gills are basically opening up and closing whenever the fish basically needs to take in water and take oxygen out of that water. In early evolution, the most anterior branchial arches, so the ones at the top, were replaced by dermal bones to form the upper and the lower jaws. Again, this is something that we're going to explore in the next coming syllabus points. Now, the last part of this syllabus point is to do with the development of the dermatocranium. We said that this is a bit more unique to the fishes, and the dermal bones, we said, come from the dermal scales, specifically from the armor fish. Okay, so the armored fish. The armored fish had basically these rhomboid scales, which is where these dermal bones come from, because these rhomboid scales basically ossified, became harder, and then made the dermatocranium. So the dermatocranium, it basically protects the skull, the brain, so it makes up the skull roof, and then it also protects the face area, so it makes up the facial bones. And it provides protection and support for the neurocranium and the visceracranium. Okay, so when you're talking about the dermatocranium, just remember the picture here. Okay, so the dermatocranium was the largest structure within the fish, and it's basically all protecting the neurocranium as well as the visceracranium because it comes before it. Okay. All right. So the maturation. So cartilaginous structures will ossify over time to become bones in order for us to support life on land. The bones of the skull will go through fusion and remodeling in order to achieve their final form. So this is a consistent thing that will keep happening. And then the soft tissues, like the muscles and the blood vessels and the nerves, are later going to come in between the bony structures in order to support lots of different various functions. So we are now done with the skull. Does anybody have any questions before we go to the last point? No? Okay. All right, then we are going to the last point of today, which is to do with the development of the skeleton. Okay. So the skeleton is made out of two parts. Two parts. We have the axial part and the appendicular part. So the axial, if you have difficulty trying to understand or remember what the differences are between them, then just remember that axial is the part of the skeleton that is protecting our most important organs. So the skull is part of the axial skeleton and it protects the brain, right? Then we have the vertebral column, which is also part of the axial skeleton, and it's going to protect the spinal cord. We have the ribs, which is also part of the axial skeleton, which is going to protect a lot of our internal organs, so like our heart, our lungs, our liver, etc. Okay, so the appendicular skeleton now is everything else. So basically our long bones, so basically like our upper limbs, our lower limbs, our pelvis, all these other things are the appendicular skeleton. So we're first going to talk about the chordata. So chordata is named chordata because as we said, the chordata, all of them will have a notochord, and this notochord is later going to become the vertebral bodies. It's going to become the vertebral column, and because of that, because the notochord is called, the notochord or the chorda dorsalis, the chordata is named after it. Okay. So this notochord, basically it is ventral to the neural tube, okay, meaning it's just in front of it, in front of the neural tube, and then it's behind the digestive system when we're talking about the embryo. It provides support It helps with cell-to-cell interactions where, the cells are telling each other where to go and what to become in embryonic induction. And it will form the neural tube, and it will basically tell the mesoderm to become parts of the cartilage. Now, the notochord is elastic, it's unsegmented, it's a longitudinal axis that comes from the mesoderm. Okay, so these are all just quick facts to know about the chorda dorsalis, or the notochord. Now, why are we mentioning this? Well, we are mentioning this because the first vertebrate that we're supposed to be talking about in evolution is the agnata, which are also called jawless fish or cyclostomes. All of these three names are the same thing, okay? So these jawless fishes, okay, they don't have a vertebral column that is very noticeable, okay? Instead, they have like some cartilage around the area where the notochord would be, but that's it. But because of these cartilage structures that they have around where the notochord would be, because they have that is why they are still considered to be vertebrates. So as these vertebrates evolved, the notochord basically became more developed and defined vertebral column. Okay, so now the vertebral column is different in all these animal groups, but they all have the same function, which is to support the body, to basically protect the spinal cord, to help with movement and flexibility. Okay, now the fishes, we mentioned this before. Fishes they have a immotile head, okay? The connection between their skull and the vertebral column is basically immotile. They don't really have like a strong connection there. And the reason for this is because fishes, they have like a streamlined shape, which allows them to swim in water very easily, okay? And you have to think of it this way, like they can also become prey. And because of that, they need to get away from their predators very easily. Or, on the other hand, they could be predators and they need to catch up to their prey very quickly. Now, if they, for some reason, had the ability to move their head up and down, all this would do is it would cause resistance in the water. So they wouldn't be able to swim properly. So because of that, the fishes, they don't have that connection. Okay, and they have a gill. They have gill apparatus, which basically makes it so that they have a very narrow kind of neck, and this would mean that their neck would not be able to would not support that movement of moving their head from like side to side or like up and down. So the vertebral column in fishes is very simple. Okay, so it has two regions. We said that they don't really have that connection between the skull and the vertebral column. Because of that, they're not going to have like the cervical region that we have, and so on. So instead, they have two parts, regio dorsalis and regio caudalis. Now, what are these two parts? Well, regio dorsalis basically means the back region, and then regio caudalis is the tail region. So those are the two the vertebral columns that they have, or the two regions of their vertebral column. Now, the vertebra that's in the regio caudalis, so in their tail, basically will connect to their ribs rather than the back ones, rather than the dorsal ones. Now, the evolution of ribs. Ribs were first seen in fishes to provide support and protection to their body, but in modern shark, these ribs may be reduced or gone completely. And this again is because we mentioned sharks are very large animals, and if they have extra things within them, kind of like if they have like a heavy skeleton, this is just going to weigh them down, and they won't be able to get to many places, and they won't be able to get their food, their prey basically. And for that reason, they get rid of the ribs. They don't have the ribs, or they have very little ribs, okay, because they don't really need them. It interferes. Now, the importance of the caudal region. So the caudal region is the most important part for the fish, because this is the part that is going to push them forward. It's the tail part of their body, which is going to push them forward or propel them forward in water. Now, the vertebrae in this region, they have hemal arches. So the vertebrae, so when you think of like the actual structure of the individual vertebrae, like the bone, they have hemal arches on them, and they're going to support the blood vessels, which are going to help in that locomotion, because those blood vessels are of course going to carry the oxygen and the nutrients that those muscles need the fish to be able to swim properly. Okay, now the evolutionary changes in the vertebral column, ribs, and sternum when it comes to the amphibians. So we mentioned this before. We said that the amphibians were the first to develop the atlo-occipital joint, atlanto. Okay, basically the joint between the atlas, which is the C1, okay, and the occipital bone, okay. And because of this, they were now able to move their head a lot more, right? And they were specifically able to move their head up and down, allowing them to catch prey very easily, or flying insects or things like that. The amphibians were also the first to develop the 4 legs. So they were the first to have, like, you know, the 2 upper limbs and the 2 lower limbs. And because of this, they had to develop the sacral vertebra, which would connect to the pelvic girdle, which would give them stability and support. The pelvic girdle is basically the pelvis, and then the pectoral girdle also. So they have a pectoral girdle, which is at the chest, right, to help them basically move their arms, to provide support for their arms. Then they have the pelvic girdle, which is going to give them support for them to be able to move their legs. Now, because of this, the vertebral column in the amphibians is a little bit more advanced than it is in the fish, because now they're going to have the regiocolli, because they have that connection between the neck and, not the neck, but the atlas and the occipital. Then they're going to have the regiodorsalis, just like the fish do. Okay, they also have the regiodorsalis, and their regiodorsalis is going to carry their ribs. And then they're going to have the regiosacralis, which is going to be important for basically helping that pelvic girdle area so that they can have support for their legs. And then they're going to have a regiocordalis, so their tail region. So when it comes to the differences between the amphibians and the fishes, when it comes to their vertebra, the difference is here. So the difference is in the fact that the amphibians have 2 extra things, the regiocolli and the regiosacralis. Okay, now in frogs and toads, the caudal region has fused into an unsegmented bone, which is called the urostyle. And this basically helps them so that they're able to be stable whilst they're like jumping or after jumping or swimming or things like that. Now, the changes in the ribs is the most interesting part for the amphibians, or about the amphibians, in that the early amphibians had very massive ribs. And this basically supported them so that they would have support against gravity. But now the ribs are much smaller, or maybe not even present at all, because the early amphibians were very big creatures. But now the next amphibians, the modern amphibians, are very small. Okay, so a lot of the structures that they would have needed otherwise, they don't need now. The sternum also developed. So this is the breastbone, right? And it basically developed coming from the pectoral girdle that I mentioned earlier. The pectoral girdle is that area in the chest that is supporting the upper arms, right? Or the upper limbs, I should say. So basically the arms. The sternum then provided essential support that the amphibian would need for quadrupedal locomotion, basically meaning that they'd be walking on all 4s. So it basically helped that ability for them to be able to walk on all 4s when they started living on land. And this again helped them transition from living in water to living on land. Now we've got the reptiles and their differences. So the cervical region of reptiles became more intricate, became more interesting, because their neck became more flexible and more mobile, so they were able to move their neck a lot more. Now, the second vertebra, so the C2, otherwise known as the axis, it basically developed the odontoid process, which will go into the ring of the C1, or will go into the atlas. And what this did was, this allowed for the reptiles, for the first time, to be able to move their head laterally, basically move their head from side to side in like a no motion. And this gave them the full mobility. So now they're able to do everything with their head, basically. So the dorsal region also, it split into 2 new parts. It split into the regiothoracis and the regiolumbalis. So instead of having a regiodorsalis, they have regiothoracis and regiolumbalis. And the reason for this is to support the thorax now in respiration, because now the reptiles are really, they have the intercostal muscles now. the intercostal muscles appeared for the first time in the reptiles and other amniotes, but not in the amphibians, because as we said, amphibians didn't really have like ribs. They didn't really need them, okay? But the reptiles do. So now they have these 5 parts of their vertebral column. So the regiocoli, of course, which allow them that full head movement, the regiothoracis, the regiolumbalis, the regiosacralis, and the regiocaudales. Now, the ribs and amniotes would need a new function, or they'd basically get a new function, which would help them in their lung ventilation because of the intercostal muscles basically would help them with their lung ventilation. So basically bringing in oxygen into the lungs. And that is why they formed the two separations, the regiothoracis and the regiolumbalis. All amniotes, they have well-developed ribs, specifically in the thoracic region, and this is to help them with efficient breathing and lung expansion. Now, the evolutionary changes in the vertebral column, ribs, and sternum when it comes to the mammals. So mammals have a sacral vertebrae which fused together to form a sacrum, and this provided stability and support to the pelvic girdle. Okay, so here we have this area right here. It's kind of short here, but right here, this is our sacral vertebrae. We also, for the first time, developed a diaphragm, which is not seen in other animals. And this was basically to help us again with our lung ventilation. Okay. And then in some animals have lost the sacral region. These animals are whales and manatees and dugongs. So basically, these are a mixture of some land animals and some aquatic animals. So they lost it because of the secondary aquatic lifestyle that they have, and because of the fact that they need to swim. So now the sacral region, we said it's going to basically be supporting the pelvic girdle, which of course is in turn supporting our lower limbs. So animals which don't really have legs or animals which are not really like walking on bipedal, so they're not like walking on their legs on the the hind legs, or they're not walking on all fours, they wouldn't need that sacral region. So that's basically what I'm saying here. Now, the structure of the sternum. So the sternum, it's got 3 different parts to it. Of course, it's got the manubrium, the body, and the xiphoid process. And the manubrium, it gives the attachment site for the clavicles, as we can see here at the top. And then the body, it articulates with the ribs, as you can see here, the blue area right here. And then the xiphoid process is like the cartilaginous extension right here at the bottom, which is the attachment points for the muscles and the ligaments. So the sternum became a bit more. It didn't appear for the first time in the mammals because the sternum appeared for the first time in amphibians, okay? But it became a bit more developed in the mammals. Now, the fusion of the caudal vertebrae in hominoid primates, so basically apes and humans. The caudal tail vertebrae would fuse together to form the os coccyx or the coccyx. And this basically led to the reduction or the loss of the tail, which is a characteristic feature of all or most hominoids. So basically, we developed the coccyx, and we don't have that caudal vertebrae that we would have had that would have supported a tail, right? Humans have 5 sacral vertebrae that fuse together to form the sacrum. This is more than in many other animals, and these few sacral vertebrates will provide stability and support to the body, specifically if we're standing in like a vertical position where we are bipedal. So humans are bipedal, meaning of course we stand upright and we walk on our 2 legs, rather than being quadrupedal, which means that we would be walking on all 4s. So if we were walking on all 4s, then the need for having the 5 sacral vertebrates kind of fusing together would not really be there. But it is now because, a lot of our force, a lot of our weight and a lot of our force basically goes onto our legs. So we need that extra support. Now, the fins and fishes. So now we're talking about the appendicular skeleton. So the fins in fishes are the stabilizers during swimming, and you can either have the paired ones, so the pectoral, which are at the front, and the pelvic, which are at the back. And then you can have the unpaired ones, which are the dorsal and the anal. Now, the dorsal one is kind of like, that fin that you see in sharks, like like in the movies or whatever, like you can see like when the shark is starting to like surface, you see that like very categoristic fin, which sticks out. That's the dorsal fin. The unpaired fins are phylogenetically older, meaning that the earliest fishes, they only had unpaired. They usually only had unpaired fins, right? Meaning that they didn't have fins on either side of their body, just maybe the dorsal one and the caudal one, and that would be it. And you can see this specifically in the jawless fishes that I showed you guys before. And them basically developing this is helpful because they were then able, they basically were able to develop this because of the signals that they sent from, and the signals that they got from the notochord. That's how they developed their unpaired fins. The paired fins are a newer innovation, so that it doesn't make sense to say newer innovation, but a newer development. The paired fins are a newer development or a innovation in vertebrates that have jaws, and they're more complex to form as they are further away from where the notochord is. So basically the notochord is that area that's going to dictate, okay, these cells need to go there and these cells need to go there, and you need to do this and you need to do this. So it's harder, or it was harder for the fishes to form the paired fins, so the fins that were at the front and the fins that were at the back, rather than the fins that were completely like towards the end of the fish's body. Those fins were easier to make, so the unpaired fins were easier to make than the paired fins. Okay, now the transition to limbs in tetrapods. So in amphibian ancestors, the paired fins would then evolve into the pentadactyl limbs that we spoke about. Remember, we spoke about the lobe-finned fishes and how they had like their fins basically kind of resembled like fingers, the 5 digits, right? So from the fishes, the amphibians got their limbs, or they were able to develop their limbs as these lobe-limbed vertebrates transitioned to terrestrial environments, so the land. The relative sizes and the positions of these skeletal elements then changed. Okay, their limbs became stronger and more adapted for supporting the animal's body on land. And then the major skeletal elements of the tetrapod limbs are the humerus, the radius, the ulna, the femur, the tibia, and fibula, which were all bones which were already found in the lobe-limbed fishes. Okay. Now, we're at the final part. So the development of the axial skeleton. Do I have a development of the... Yeah, that's it. Okay, yeah. So the development of the axial skeleton. So I don't have something on the development of the appendicular skeleton because I don't think that you need to know that. I mean, we've already spoken a little bit about it. We have spoken about the evolutionary changes, but we haven't really spoken about the actual development of it. But I don't think that was something that was mentioned in the lecture slides, so it's okay. But the vertebrae, so the vertebral column, basically develops bit by bit by segmental deposition of cartilage. So we're going to start off with cartilage, of course, before we later ossify it and we later make it into bone. Okay, so sclerotomes, which are parts of the somites, they're middle parts of the somites, they have a very important role in this process. These sclerotomes, you can see them right here, are positioned adjacent to the notochord. They're like basically right next to the notochord. And these sclerotomes are going to produce cartilage. And then these cartilage cells are then going to form the bones of the vertebrate and the bones of the ribs. So the epithelial cells, which are found in the sclerotomes, are going to transform themselves into mesenchymal cells, and they're going to move around the notochord. And then each vertebrae part has parts of 2 sclerotomes, and then the caudal part of it's got a caudal part of 1 sclerotome. So the bottom part of 1 sclerotome, right? It's got that, and then it's got the cranial part of it next. I don't know if that makes sense. Okay, so basically, if you think about the vertebral column, right? So you have the order, you have, your C1, your C2, your C3, and so on, right? So basically, what they're trying to say is that the sclerotomes basically arrange themselves in a way where one of them would be like at the top, then the other one would be at the bottom, and then one of them at the top and the other one at the bottom, and so on. And then they would deposit their cartilage, basically. Now, the vertebral segmentation would facilitate the spinal nerve roots because they have like little gaps in between them, right? They have little gaps in between the sclerotome, which would allow for the spinal nerves to go around, and the peripheral nerves and so on. And then the anterior end of the notochord, so the anterior end being this part, is incorporated into the base of the skull. So it goes into the base of the skull and it's not replaced by sclerotomes. So essentially what I'm saying here is that the sclerotomes would replace parts of the notochord. Sorry, the notochord is here. The sclerotome would basically replace parts of the notochord, but then the top part of this notochord would go to the top part of the skull, or the base part, I should say, of the skull. So this occipital region. So it's not replaced by vertebrae. Now the development, further more on the development. So ribs initially develop as part of the cartilage module for each vertebrae. So we have something called endochondral ossification, which you learn about in cytology. You learn about it in cytology. And basically, what we mean by this is, so you have two types. You have two ways of bones forming, okay? So you have the intramembranous ossification, which is basically the direct formation of the bone, okay? So you have basically the osteoblasts that are making the bone directly. Then you have the endochondral ossification. And in endochondral ossification, you're going to start off with like a model, the model basically being made out of cartilage. In some cases, it can be hyaline, so it might be made out of hyaline cartilage. Now, then when you have this model cartilage, what you're going to do then is the body's going to then start to produce the bone, or it's going to start to ossify the bone around this hyaline cartilage. As you know, the hyaline cartilage is the model or the shape that it's trying to become. So the ribs, they start to form as part of the cartilage model for each vertebra. So in the thorax region, the rib part separates from the vertebra and then becomes the real ribs. So at first, ribs and the vertebra are developing together, okay? They're basically becoming the same thing, they're the same thing, and then the ribs are going to separate. The ribs are going to go away from the vertebra, and they're going to make the real ribs. The sternum is going to form as a paired cartilage model on either side of the anterior midline. So basically, if you look at the structure of the sternum here, what we're saying is we're eventually going to have one part of the sternum and then the other part of the sternum on either sides of the body, and then they're going to come together to fuse together to make the sternum during embryonic development. And yes, where were we? We are here. Okay, cool. Then the cartilage models of the ribs are going to become attached to the lateral sides of the developing sternum, which you can see in the picture here. Okay, as you can see, the sternum is right here and it's articulating with the ribs right there. And then the last point of what we're talking about today, then eventually the two halves of the cartilaginous sternum are going to fuse together along the midline. So eventually, as we said, the sternum is going to come together in the middle.